Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about the Hexose Monophosphate Shunt pathway or HMP shunt pathway, which is also known as Pentose Phosphate pathway or PPP. Okay, this video is going to be useful for all the NEET aspirants, NET aspirants and UG and PG students of biochemistry, zoology, etc. Okay. So, why this pathway is called HMP pathway or hexose monophosphate pathway? See, this is a minor pathway of carbohydrate metabolism, and whatever intermediates are formed here, they are all of monophosphate type. Okay, and they are mainly formed from the glucose, which is a hexose sugar. Okay, that is why this pathway is known as hexose monophosphate pathway. Now, why is it called pentose phosphate pathway? Because see, the initial intermediates are pentose phosphate in nature in this pathway. What are those intermediates? Like ribulose 5-phosphate, ribose 5-phosphate, xylulose 5-phosphate. Okay, those are all 5 carbon containing pentose phosphate in nature. Okay, that is why this pathway is also known as pentose phosphate pathway or PPP. So now, why this pathway, this minor pathway of carbohydrate metabolism is needed? See, we know that glucose gets converted into glucose 6-phosphate with the help of hexokinase, and then that glucose 6-phosphate undergoes glycolysis process and relieves 2 molecules of pyruvate from 1 molecule of glucose. Now, 10% of the glucose gets converted into glucose 6-phosphate, but they does not undergo glycolysis. Rather, they shunts through the HMP pathway or PPP pathway. Okay. Now, why this 10% of glucose bypasses through the HMP pathway? See, there are two main reasons. One is the generation of NADPH2. Now, what is NADPH2? NADPH2 is a coenzyme. Okay, remember that NADPH2 is different from NADH2. Okay, NADH2 is that coenzyme which goes through the ETC or electron transport chain and relieves the ATP. Okay, generates the ATP, but NADPH2 generally does not generate ATP. Rather, it has other functions, other very important functions in our body. What are those functions? It helps in free radical scavenging. Now, what is free radical scavenging? See, whenever the superoxide anions are formed in our body, okay, in our cells, they gets converted into hydrogen peroxide and other free radicals which are very harmful for our body, harmful for our cells. If these H2O2 or free radicals gets accumulated into our cell will lyse after some time. Okay, now this NADPH2 saves our cell from the cell lysis driven by those free radicals. Okay, so these NADPH2 actually scavenge or takes out those free radicals from our cell in the form of non-toxic molecules. Okay, that is why it is very important for our cell survival or cell viability. Not only that, it also helps in fatty acid, cholesterol and steroid synthesis. Okay, now another thing is that this pathway also helps in generation of pentose phosphates, like I have already told you, ribose 5-phosphate, ribulose 5-phosphate, xylulose 5-phosphate, etc. Okay, now these pentose phosphates are important component of DNA and RNA synthesis. So that is how this pathway also helps in DNA and RNA synthesis. Okay, so to form the NADPH2 and pentose phosphate, this pathway is actually needed. Okay, this pathway is found in almost all cells of the body, but majorly in liver and RBC. Okay, abundantly. Now directly going to the pathway. 6. See, as we know that glucose gets converted into glucose 6-phosphate with the help of hexokinase, and it is a 6 carbon compound. Whatever I have written in the circle, that is their carbon content. Okay, carbon number of carbon moieties. Now This glucose 6-phosphate gets converted into 6-phosphogluconolactone. What? 6-phosphogluconolactone with the help of G6PD or glucose 6-phosphate dehydrogenase enzyme. This enzyme is very important and it is a rate limiting step, okay, in this reaction. So you can see this is a dehydrogenase enzyme, okay. So obviously some oxidation reduction reaction will be there. See, in this reaction, you can see one molecule of NADP gets converted into NADPH2. Okay, so NADP gets reduced into NADPH2, whereas glucose 6-phosphate gets oxidized into 6-phosphogluconolactone with the help of G6PD. Okay. Now in the second step, we can see that the 6-phosphogluconolactone gets converted into 6-phosphogluconic acid or gluconate. Okay, 6-phosphogluconate or 6-phosphogluconic acid with the help of gluconolactone hydrolase enzyme. Why hydrolase enzyme? Because you can see here the water molecule is added here. That is why hydrolase enzyme. Now this 6-phosphogluconic acid again undergoes oxidation reduction reaction and gets oxidized into a very transient form known as 3-keto-6-phosphogluconate, okay, with the help of 6-phosphogluconate dehydrogenase enzyme. Okay, so obviously here also NADP molecule gets reduced into NADH2. Okay, so here two steps, you can see the oxidation reduction reaction is occurring in this step, first step, and in this third step. Okay. Now this 3-keto-6-phosphogluconate is a very transient compound, so it gets it releases carbon dioxide and gets converted into ribulose 5-phosphate. As it is releasing one carbon dioxide, okay, so obviously the carbon number in the molecule will decrease, and that is why the carbon content of the ribulose 5-phosphate is now 5, okay, not 6. Okay, now in this reaction also the same enzyme helps, that is 6-phosphogluconate dehydrogenase. So 6-phosphogluconate dehydrogenase not only does the oxidation reduction reaction, it also does the decarboxylation reaction. And till now whatever reaction you have seen, those are all the oxidative phase of HMP pathway. Okay, now another question you may ask that, ma'am, what is these numbers 6, 6? Okay, what are these numbers? See, in the HMP shunt pathway, we start our reaction with 6 molecules of glucose 6-phosphate. So these are the numbers of how many molecules of glucose 6-phosphate you have taken. Okay, so 6 molecule of glucose 6-phosphate gets converted into 6 molecules of 6-phosphogluconolactone. Then they gets converted into 6 molecules of 6-phosphogluconic acid. Then from that 6 molecules of 3-keto-6-phosphogluconate, and after that 6 molecules of ribulose 5-phosphate. Okay, as we are dealing with 6 molecules of each and everything, so obviously in first and third step you will see that 6 molecules of NADP gets converted into 6 molecule of NADPH2. Similarly in this step also, and in this step 6 molecules of water is added, and here 6 molecules of carbon dioxide is released. Okay, now these 6 molecules of ribulose 5-phosphate, now onwards whatever reaction you will see are of non-oxidative type. Okay, no oxidation reduction reaction will occur here, no NADPH2 will now form. Okay, now from these 6 molecules of ribulose 5-phosphate, you can see 2 molecules of ribose 5-phosphates are formed and 4 molecules of xylulose 5-phosphates are formed. Okay, now both of these are 5 carbon component. Okay, now ribulose 5-phosphate and ribose 5-phosphates are isomer of each other. Ribulose 5-phosphate is a keto sugar, whereas ribose 5-phosphate is an aldose sugar. So the enzyme which converts them or isomerize the ribulose 5-phosphate into ribose 5-phosphate is phosphopentose isomerase enzyme. On the other hand, xylulose 5-phosphate is an epimer of ribulose 5-phosphate. Okay, it is also a keto sugar, but it is an epimer of ribulose 5-phosphate. So here the phosphopentose epimerase enzyme converts it into xylulose 5-phosphate. Okay. Now you can see 4 molecules of xylulose 5-phosphate are there and 2 molecules of ribose 5-phosphate are there. Now these 4 molecules of xylulose 5-phosphate gets converted into 2 molecules of xylulose 5-phosphate and another 2 molecules of xylulose 5-phosphate. Now these 2 molecules of xylulose 5-phosphate reacts with these 2 molecules of ribose 5-phosphate with the help of transketolase enzyme, where TPP or thymine pyrophosphate acts as an coenzyme, and they form 2 molecules of glyceraldehyde 3-phosphate and 2 molecules of pseudoheptulose 7-phosphate. Okay, see here we have started with 5 carbon and 5 carbon, so 10 carbon total, and we are now getting 3 carbon glyceraldehyde 3-phosphate plus 7 carbon pseudoheptulose 7-phosphate. So total 10 carbon are made here. Okay, now these 2 molecules of glyceraldehyde 3-phosphate and 2 molecules of pseudoheptulose 7-phosphate again reacts with each other with the help of transaldolase enzyme and form 2 molecules of erythrose 4-phosphate and 2 molecules of fructose 6-phosphate. Okay, erythrose 4-phosphate is 4 carbon compound and fructose 6-phosphate is 6 carbon compound. So 10 carbon is made here also. Okay, now these 2 molecules of fructose 6-phosphate is directly goes into the next step. Okay, now these 2 molecules of erythrose 4-phosphate now reacts with these 2 molecules of xylulose 5-phosphate which have not reacted till now. Okay, so it has 5 carbon, it has 4 carbon, so total 9 carbon. So these 2 molecules reacts with the help of transketolase which have TPP coenzyme, and they form 3 carbon containing 2 molecules of glyceraldehyde 3-phosphate and 6 carbon containing 2 molecules of fructose 6-phosphate. So 3 plus 6 9 carbon is made. Okay, and these 2 fructose 6-phosphate is directly goes to the next step. Okay, and these 2 molecules of glyceraldehyde 3-phosphate which are 3 carbon in nature gets condensed into 1 molecule of 6 carbon fructose 6-phosphate, and it is a reversal reaction of glycolysis. As in glycolysis, we have seen that 1 molecule of fructose 6-phosphate gets converted into 1 molecule of glyceraldehyde 3-phosphate and 1 molecule of DHAP with the help of aldolase. Here the reverse reaction occurs where 2 molecule of glyceraldehyde 3-phosphate gets condensed into 1 molecule of 6 carbon containing fructose 6-phosphate. Okay, now how many fructose 6-phosphate we have got till now? 1 plus 2 plus 2, 5. Okay, so these 5 fructose 6-phosphate goes to the next step. These 5 fructose 6-phosphate goes to the next step. Fructose 6-phosphate are all 6 carbon containing molecule. Now from this fructose 6-phosphate we can get glucose 6-phosphate. It is also a reversal reaction of glycolysis where phosphohexose isomerase enzyme reacts. Okay, or helps in this reaction. So from 5 fructose 6-phosphate we can get 5 molecules of glucose 6-phosphate. Glucose 6-phosphate are obviously 6 carbon containing compound. So overall we can see that we have started with 6 glucose 6-phosphate and at the end of the reaction we are getting 5 glucose 6-phosphate. So 1 glucose 6-phosphate is completely oxidized in this reaction. Okay. So in summary, we can say that 1 glucose or 1 glucose 6-phosphate, whatever you can say, is completely oxidized, and 5 glucose or 5 glucose 6-phosphate are regenerated at the end of one cycle of HMP pathway. Okay. And for complete oxidation of 1 glucose molecule, we can get 6 carbon dioxide. 6 carbon dioxide are eliminated. See here in this step. In what step? In this step we can see 6 carbon moieties of single glucose 6-phosphate molecule is liberated in this step. Okay, so 6 carbon dioxide molecules are eliminated. And our primary goal was to generate NADPH2. Okay, so in a single cycle we can see 12 molecules of NADPH2 is generated. Why 12? Because see here, in the first step 6 molecules of NADPH2 was generated by oxidation reaction, and in the third step also by the process of oxidation reduction reaction 6 molecules of NADPH2 is generated. So 6 plus 6, total 12 molecules of NADPH2 is generated. Okay. And another interesting thing is that no ATP is either consumed or generated in this shunt pathway. Okay, that is the main difference with glycolysis. In glycolysis, we can see in aerobic glycolysis 7 molecules of ATP are generated. In anaerobic 2 molecules of ATP are generated. But here no ATP is either consumed or generated. Okay, so it is not a ATP dependent process. Okay. Now, this is all about the HMP shunt pathway. Now, coming to its regulation. At the very beginning, I have told you that G6PD, which does the first reaction of this cycle, that is the conversion of glucose 6-phosphate to 6-phosphogluconolactone, G6PD is required. Okay. Now, this G6PD was the rate limiting enzyme. Okay, so this G6PD regulates the rate of this whole pathway. How? See, this G6PD is actually inhibited by the NADPH2. Why? See, this we are running this reaction, or we are running this pathway for the generation of NADPH2. So if you have ample amount of NADPH2 in your hand, so you don't need to run this pathway, right? So that is why the excess amount of NADPH2 actually inhibits the G6PD enzyme, so that the overall pathway is inhibited. Okay. On the other hand, if you have less level, if you have higher level of NADP plus in your body, you will try to convert it into NADPH2. Okay, so the level of NADP plus actually regulates if the pathway will occur or not. Okay. Now another thing also regulates the oxidative phase of this HMP shunt pathway, that is insulin. Insulin actually induces the G6PD. Okay, so insulin helps in induction of G6PD. As a result, the overall pathway is induced. Okay, so insulin helps in this pathway. On the other hand, the non-oxidative phase, okay, afterwards the formation of ribulose 5-phosphate, whatever reaction we have seen are those are all in non-oxidative phase. So the non-oxidative phase actually controlled by requirement of pentose in our body. If we have ample pentose in our body, the non-oxidative phase will not run, and if we have dearth of pentose in our body for formation of DNA and RNA, then obviously the non-oxidative phase will run. Okay, so that is how the regulation of this pathway occurs. Now, the most important thing about this lecture is the clinical significance of G6PD and obviously the pathway. See, I have told you G6PD is the rate limiting enzyme here, and G6PD helps in obviously the production of NADPH2, because if the G6PD is not working, the pathway will not occur and the NADPH2 will not form. So G6PD helps in the production of NADPH2. And this NADPH2 in turn helps in free radical scavenging. Now what is this free radical scavenging? Let's understand here. See, in our cells, when the oxygen is in ample amount, the oxygen gets converted into its superoxide anions. Okay. Now, whenever superoxide anions are formed, they perform chain reaction and form other free radicals. Superoxide anions are itself a free radical, and in the subsequent chain reaction, they form other free radicals which are very harmful for our cells. See here, with the help of superoxide dismutase or SOD, the superoxide anions form H2O2 or hydrogen peroxide, which is also a free radical and very harmful for our cell. Now, obviously, our cell will try to get rid of this hydrogen peroxide. Okay, so our cell actually converts this hydrogen peroxide into non-toxic water. Okay, with the help of glutathione peroxidase or POD enzyme. Now for the functioning of this glutathione peroxidase enzyme, we require reduced glutathione. What do we require? Reduced glutathione. Remember this. Reduced glutathione is required for the proper functioning of glutathione peroxidase. Okay. Now in this reaction we can see the glutathione gets oxidized, okay, and the H2O2 gets reduced into water. Now to keep this reaction on, we need to convert this oxidized glutathione into its reduced state, obviously. That is when this reaction can occur. Okay. So this oxidized glutathione gets converted into its reduced states by the help of glutathione reductase enzyme. And in the performance of this glutathione reductase enzyme, our NADPH2 helps, which we have got from the HMP shunt pathway, okay, with the help of G6PD enzyme. So in conclusion, we can say that through the HMP shunt pathway, whatever NADPH2 we have got, they helps in the functioning of glutathione reductase. This glutathione reductase enzyme is necessary for converting oxidized glutathione into its reduced state, okay, and this reduced glutathione in turn helps in the conversion of toxic H2O2 into non-toxic water molecule by helping glutathione peroxidase enzyme. So that is how NADPH2 helps in keeping out our cell from the toxic elements or toxic free radicals, okay, and that is how they helps in preventing the cell lysis. If H2O2 gets accumulated in our cell in large amount, our cell will get lysed or our cell will not be viable. So that is how NADPH2 helps in reduction of oxidation of H2O2 into water, and that is how they save our cell from cell lysis. So NADPH2 is very important for free radical scavenging and for our cell viability. Now see, this G6PD enzyme, which helps in the formation of NADPH2 through the HMP shunt pathway, is located in our X chromosome, okay, and if there is any recessive mutation in that particular gene of G6PD, then we may lose the function of G6PD enzyme activity, okay, and if the G6PD enzyme activity is lost, obviously less amount of NADPH2 will be formed or no NADPH2 will be formed, okay. As a result, the hydrogen peroxide will not be oxidized into non-toxic water, okay, and as a result the hydrogen peroxide will accumulate in our body and our cell will get lysed, okay, our cell will not be viable. And in case of anti-malarial drug primaquine, we can see this kind of state, okay. See, if a person is infected with malaria, then he needs to take anti-malarial drugs like primaquine. But the problem is that these drugs actually helps in production of H2O2, which is very harmful for our cells. Now, see, if that person is G6PD deficient, already G6PD deficient, he is also deficient in NADPH2. So these H2O2, these increasing amount of H2O2 will not get converted into H2O, as the person is lacking the NADPH2, right? So there will be no free radical scavenging. As a result, H2O2 will accumulate in our cells, and after that cells will get lysed, and these kind of things are known as drug-induced hemolytic anemia. Why? The primaquine drug has induced the formation of free radicals, and that is why the G6PD deficient persons are facing the hemolytic activity of RBC. Okay, the lysis of RBC cells, and as a result they are suffering from anemia. That is why this is known as drug-induced hemolytic anemia. But another thing is that, see here, the G6PD deficiency has also some benefit, and our nature has made a beautiful thing out of it. See, wherever the forest cover is there, tribal peoples remain there, right? And in the forest we know that various mosquitoes are there, okay, and the mosquitoes are vector of plasmodium, okay, the microorganism responsible for malaria. Now see, these plasmodiums needs reduced glutathione or GSH for their survival, okay. So the plasmodium, the malarial parasite, needs this reduced glutathione for their survival. Now in the forest area, people are very prone to get malaria, okay, because there are several mosquitoes are there. So in those tribal communities, we can see nature have framed in such a way that those people are actually G6PD deficient, okay, their G6PD level is very low. As a result, their NADPH2 level is also very low, okay, and if your NADPH2 level is very low, obviously the level of reduced glutathione will also get lower, okay, because the oxidized glutathione will not get converted into reduced one. So reduced glutathione will also get lower. As a result, the plasmodium may enter into their cells, but the plasmodium cannot survive in their body because they require higher amount of reduced glutathione for their survival, okay. But if you are already G6PD deficient, your reduced glutathione content is already less, so the plasmodium cannot survive in your body, okay. So that is how in the forest area the tribal communities are naturally protected from malaria, okay. And in the forest covered states of India and Africa, we can see this kind of G6PD deficiency which actually protects those tribal people from getting malaria, okay. So that is how G6PD deficiency has a benefit over malaria disease, okay. So I hope this video is very useful for you for your examination and you have understood everything very clearly. If you have any doubt, you can comment down below. I will answer the questions, okay. And if you find this video useful, don't forget to comment it below, okay. And obviously share it with your friends and subscribe to my channel for more such videos. Thanks for watching and happy learning.